Well, good evening, everyone. It is my pleasure to introduce Cordelia Running, who is our speaker for tonight, and I'm going to let Cordelia introduce her topic.
Well, hello. Thank you so much for having me. And I'm happy to share with you this evening some of the work that's been done by my lab over the few years and some of the ideas we've been testing out related to how people experience flavors from their food in particular. If anyone has any questions, you're always welcome to go ahead and ask, type something in the chat. You don't need to be formal, but also happy to chat at the end as well. So, all right. So the things I wanted to share, I was going to focus on what I'm calling functional saliva assays. They're basically some creative stuff I hope they're creative things we kind of invented as a result of the COVID era here, that we're trying to connect back to the way people experience their food and then trying to see if we can use that to track whether people are shifting their experiences of their food. And so I like to start off with the acknowledgements because that way, if I talk for too long, we still get to this part, and that because it's important. And so a lot of the students who did work on these projects are shown here on the screen. Several master's students and PhDs. So I've got to add some PhDs and some master's notations to several of these folks. And then we've had some funding from internally from Purdue University, as well as some support from USDA and NIH for some of the projects here. And I do some consulting on occasion, but nobody had any influence on any of this work. Love to have some support for some of this work, but of course, they don't get to influence the outcomes. All right, so in my lab is called the Saliva Perception, Ingestion, and Tongues Lab, or the SPIT Lab, and that's because my last name is Running. And when you have a last name of Running, you have to invent an acronym for your lab, otherwise people get very confused about what you actually do. But in general, our goal is this idea that food should taste good, even when it's healthy for you, and even when you aren't healthy. But the problem with that is going back all the way to Confucius, or more recently to my dad, good medicine tastes bitter. And this is actually true. It's not just an allegory of many things in life, but things like ibuprofen taste bitter, and things in food that are good for you, presumably, taste bitter as well. So things like minus epicatechin, which is one of the compounds in chocolate we think might contribute to its potentially positive cardiovascular effects, if you don't eat too much of the fat and the sugar that tends to come with the chocolate as well. And the other thing is that many of these same chemical stimuli being responsible for both the positive health consequences as well as these unfortunate flavors kind of creates a problem. And so this is true for all sorts of compounds that have been studied in foods, from alloisothiocyanate, which is found in things like garlic and onions and mustards, and it's a lot of the burning sensation you might get from those. Capsaicin has been investigated for various medical or therapeutic properties, but obviously has a spicy or tingling or burning effect. And then even things like the quinine in tonic water. So quinine in tonic water originally was a tonic, or originally was used for its health benefits or medicinal benefits. And yet now we use the same stimulus, the same chemical molecule. We continue to use it in things like tonic water for the pleasure, so to speak, of drinking that beverage, even though it's bitter. And so if you're hearing the beeping of my computer, I'm sorry, I keep pushing the next arrow and it doesn't like that for some reason. And so again, my lab investigates sort of how we can get people to like these healthy flavors that are not necessarily inherently likable. And we do that through looking at these interactions of saliva with flavor and with diet and people's experiences. And so when we think about flavor, a lot of the sensations that we're interested in here are going to be things like bitterness and fattiness in the texture of foods. When we think about diet and experience, your recent or habitual diets might influence saliva, and it might influence the flavors you experience. And certainly the experiences we've had culturally, personally, with our families through our eating habits and behaviors influence our acceptance of flavors. And then saliva, I love this phrase. Unfortunately, I didn't come up with it, but saliva is the unavoidable ingredient in everything that we eat. And so it's this chemical component that is added to absolutely everything you're going to consume. Even if you're fed through like a tube that goes into your nose down into your belly, you're constantly swallowing saliva all day long. Somewhere between 1 liter to 1.5 liters is the estimate of how much of your own saliva you drink in a day. And so it's not nothing when we start to think about the amounts of this that actually end up in your food, in your gastrointestinal tract. And so thinking about the way these things interact is really sort of the science foundation of how my lab is looking into these patterns. And so introducing this other concept of these functional saliva assays, the whole goal here is to be able to measure some of these aspects of saliva related to the flavors people experience and their diet and what they think of those flavors. And so I've got 3 in particular that I'm going to talk about today. One is how saliva might stabilize mixtures of oil and water, and how it might influence people's experience of the flavor of oleogustus, which is the taste of fatty acids in foods. And then I'm going to talk about some measurements we've done related to salivary digestion of starch, which is probably the most common thing that people think about when they think about sort of some of the functional properties of saliva and what it does to food. And then finally thinking about how saliva has some components in it that will bind bitter or astringent stimuli in foods. And starting off with this idea of this saliva stabilizing oil and water mixtures and potentially influencing fatty acid taste. Yeah, that's the one we're going to start with here. So again, I just want to, if you don't sit around and think about triglycerides very often in your regular life here, I figured I would remind you that fatty acids, they're a component of triglycerides. So triglycerides is the fat that you find in foods, generally speaking. And the fatty acids, which are just a part of this molecule, are what we actually think is the stimulus for fatty taste. So there actually is a taste sensation from the mouth, from taste buds that are present in the mouth, that comes from fat. But it's from these fatty acids, not the whole triglyceride molecule. And so when we look at a triglyceride, the way you end up with the fatty acids is this ester bond gets cleaved, and you end up with individual fatty acids that can be broken off of the triglyceride, right? And so again, these fatty acids, they can get released through the activity of lipase. So lipase is a type of enzyme that will come in and it will cleave that bond. It's naturally present in a lot of foods, especially dairy products. It's also present in very small amounts in your saliva, but it is there. And then also if the fat is treated with acid or base and heat, these bonds can break apart, these ester bonds can break down. And so in general, you can just naturally have fatty acids present in the food. Generally not a ton of them though, because most people don't find them very pleasant tasting. And we'll get to that in a moment. So again, fatty acid breaks off the larger fat molecule. The lipase can come from the food itself or from your saliva. Acid and base hydrolysis, you can intentionally release more fatty acids that way. And you can also cause hydrolysis through heat. So for example, when you fry things in oil, you can end up with more and more free fatty acids, non-esterified fatty acids being released from the triglyceride. That's one of the reasons why fryer oil goes bad eventually. So some of that smell you get from old frying oil, some of that smell you can trace back to the smells of these fatty acids as they break apart. And so we wanted to know if a person's experience of the taste, so very specifically the taste of those fatty acids, oleogustus, if that would relate to their saliva's ability to mix fat and water. Because as I has been studying this particular taste over the years, some of the things that I'm always struck by are, just how exactly are these very lipophilic things actually accessing receptors present in the mouth, which is coated in saliva, which is mostly water. And so we wanted to see if there might be some like effects of the emulsion itself going on here that might relate to how people experienced taste of fatty things. And also if that's stable over time, though we don't have much to report on that front yet. And so what we did with this study is we had people swish and spit out an oil and water mixture. And you might think, oh, well, why didn't you do a lot of analytical measures of all these things? And well, if you look at the date, we got this published finally in 2022. So most of this work was happening in 2021. And so, being somebody who studies saliva in the COVID environment was not a great time for my research lab, but we adapted. And so we actually did all of these things virtually. We had people come in and pick up kits and then do it through a Zoom call with them. And we took pictures in order to make our measurements. And so people would swish this mixture of oil and water. It had vegetable oil, which is going to be the clear layer in a lot of these images you're about to see. And then we added some purple food coloring to the water. And so that would separate out to the bottom in the original oil and water mixture. We have people drink the mixture into their mouth, swish it around their mouth for 30 seconds, spit it back out into a tube, into a vial, and then we observed the layers that were formed. And they're actually quite striking. So this is the original. This is before somebody would have sipped this into their mouth. And again, you see just a very clear vegetable layer on top, vegetable oil layer up top here, and this very purple water layer below. And after people would swish with this is the kind of thing you would see that they're spitting out. So immediately, especially when I talk to chemists, it's like, well, what on earth is going on with the partitioning here? Clearly, there's some pigment that wants to shift into that upper layer, and then the color of the lower layer of the water seems to shift as well because of the loss of that sort of pinkish pigment. So lots of cool stuff going on here that we're now measuring now that we, are back in the lab a bit more. And again, most of that is related to the food coloring that we happened to pick that worked really well for this. But it made it really easy to see how well people's saliva was influencing these phases. And so I'll show you some examples about that. And then also we related, how this sort of emulsion was forming and how it was stabilized over time back to people's experience of fatty foods that we had them taste. And so this is kind of the kit that we sent home with people. We tried to use some sort of color correctors in the kit, but the lighting, we needed to like make a light box basically for people to be able to do this really well. But it was, we learned a lot in our first time around here. But this is sort of the setup they would do. So everybody had a control that they just left alone. We had taped this shut so that they can't mess with it. And then they had a second vial, which was what they actually sipped into their mouth, swished around and spat back out. And so this is a spat out sample from someone. And so we've got this upper layer that kind of goes pink. Again, these were the same thing before it went into somebody's mouth. So clearly there's something about the saliva that is changing the way these pigments are partitioning. And it's also really different amongst the people that we tested. Right. And so to show you that, the measurements that I'm going to tell you about here in a moment, to give you some context on that, basically we're just doing a ratio because again, all of this was done virtually. We did what we could. We're working on some more, fancy measurements of this to, actually figure out what's where and if we can make it more precise and things. But just as a rough initial measurement, we just counted the pixels in the images and made ratios of the size of the upper layer. So in theory, this is the fattier layer, the cream layer, so to speak, compared to the overall volume, the overall like height of the sample. To give us an idea of the ratio of this whole sample that seemed to be the fatty layer. Because the larger that layer is, the more evenly mixed the water and the oil are. And so up here, this first image, this is an example of somebody who had very unstable mixture. So again, they swish this around their mouth for 30 seconds, they spit it into the tube. And at the beginning, you can see there's already a bit of a water layer down here at the bottom of this tube. And after 3 minutes of the tube just sitting and taking repeated pictures, we see a very clear separation of our fatty and our watery phase. Interestingly, that pink pigment stays in the top layer for a really long time. So that was interesting to watch. But then again, you can see that upper layer shrinks over time when someone has an unstable mixture they're spitting out. Versus another person who, if you look at these images, there's almost no water layer at all. You can just see it starting to form here. At 1 minute, there's a tiny one maybe, and then at 3 minutes, you see this tiny, tiny little purple layer starting to show up at the bottom of the image. And so again, this is the same sample. They've been given the same directions. We did some experiments in the lab about, well, maybe they're just swishing really, really hard if they're this person. And so we tried like shaking it really, really hard versus shaking it just a little and seeing if that influenced the way these phases were mixing. And it really didn't. It really didn't matter that much. Like it would separate if it wanted to separate, and it wouldn't separate if you had somebody with this different saliva that seemed to really emulsify the two phases well. And so that was really interesting in and of itself. But then we were also able, right click going here. So again, these are just, the arrows are just to, again, emphasize to you that there's very little of that water layer forming for this person. We were also able to relate then these sort of proportions of how much of this upper fatty layer was present in these mixtures to people's experience of that fatty acid taste. And so we had given people a bunch of samples. We ruined chocolate. We took some white chocolate. It's not technically chocolate. It's that melting chocolate kind of stuff, the coating that you can get at the grocery store that you don't have to temper, because it was easier is why we used it. And we spiked it with some fatty acids, which makes it taste terrible. If you've never come across this term before, don't think of this as like, ooh, oh, fatty taste. That must be nice. No, it's not. The type of fattiness that you taste through this particular sensory system is not a good thing. Again, bad fryer oil. That's what we're talking about here. And so again, we spiked some white chocolate with some fatty acids, and we asked people to, can you tell them apart, right? Because some people are really good at this. They can detect these fatty acids, this sort of rancid taste really easily, and other people are just not very good at it. And it turned out that at least in our first test here, what we observed is looking at that upper layer fraction of this spat out sample, people who failed this test for trying to detect the fatty acids, they actually had smaller upper layers over the whole time course of this experiment. Well, particularly the first two time points, but in the main effect model, I think it was significant in general. But again, you see, particularly early on, these people had worse emulsions that they spat out, indicated by the fact that the size of this upper layer was smaller compared to the people who passed, who were able to detect the sort of disgusting fatty taste, right? So again, people whose spat out samples of oil and water looked more like the person on the top, these people tended to be unable to discriminate our fatty acid taste. Whereas the people whose samples looked more like the spat out mixture on the bottom, these people were more likely to be able to detect that fatty acid taste. And so that was interesting to us because, again, like this is, if this mixing is influencing the way that these components of someone's food are able to interact with taste receptors, maybe by making the better emulsion, we're getting finer droplets. Maybe they're more easily able to get to taste receptors. Maybe it's more able to have a carrier protein get onto the outside of these fat droplets and then be able to take the stimulus to a taste receptor. We don't know really what's happening yet, but it was interesting. It was pretty cool to be able, well, I mean, during COVID when you're making up new methods because you had to, it was a really interesting effect for us to see. So we're hoping to be able to use this in future studies to kind of adapt and learn some more things. So in general, from this emulsion stability thing, you know, something that always comes back to me is what is going on with that pink pigment. So I did do some work digging into the actual food coloring we used. Some of this was just getting lucky, picking the right food coloring, because it's what I happened to have in my kitchen during COVID when you couldn't buy anything because all the supply chains were messed up. And so it does turn out that one of the red pigments that's present in this particular purple color that we had picked is kind of a known binder of like everything. It's kind of an indiscriminate protein, interacts with indiscriminately with a wide variety of proteins. And so it's likely that there's some salivary protein that tends to migrate to the fatty layer that is then bringing that red pigment with it. But it does make it really easy to observe this effect. So that's actually really useful. We're hoping to do some more work looking at the repeatability of this assay, both within a person and over time. Maybe, you know, if we wanted to do a better photopox so that we continue to do these studies remotely, because that was actually really useful for being able to broaden sort of the diversity of our sample rather than only having people who happen to walk into a nutrition department on campus to do our study. We can get a lot more people and different types of people into the research study. And then in general, being able to kind of understand that fatty sensation and how exactly that is being influenced by something that is related to the stability of the oil mixing with the water, mixing with a person's saliva. That's something that I'm really hoping we'll be able to do some more work with in the future. Because again, the goal of my research is trying to get people to eat healthier foods. And hopefully one of the ways to get them to eat healthier foods is to make the healthier stuff more appealing to them. And so being able to shift those sensations, well, maybe if we understand the way the fat and the saliva are all interacting, we might be able to influence that more directly. All right, so that's the fat one. The next one I want to talk about is related to salivary digestion of starch. And so we've actually been doing this particular version of this experiment in my food chemistry class for, I don't know, 8 years now, where basically I give students in the class a cup of pudding that we buy off the shelf. So this is just shelf stable pudding you can buy at the grocery store. They're like $1 for the 4-pack. And what I always had them do in class is they take a picture of the original pudding and then they lick the spoon. So you don't even like fully spit into it. In this case, you just lick the spoon and then you stir it for a while. And this is what happens. You go from a very thick pudding to it slowly turns into chocolate milk, or quickly for some people. And the amount of difference within the classroom that I was observing, I was very surprised by it and was thinking, all right, maybe we can turn this into something to measure in the research environment. That's very inexpensive, it's very portable. And so we kind of adapted some other methods to create our own version of being able to measure this sort of viscosity change in saliva or in pudding by the addition of somebody's saliva. Right. And so kind of as a background for that, saliva contains alpha amylase. Right? It hydrolyzes the alpha 1,4 glucose bonds of the amylose fraction of starch. So if you're not familiar with starch structure, starch contains amylose, which is going to be this chain up at the top, and amylopectin portions within the starch. So amylopectin portions are going to be the branched ones. So they go off the 6th carbon in the glucose ring. So kind of dangling off the tail off to the side. And so these are branched kind of really dense regions of the starch molecule, whereas these amylose sections are these long chains. And again, this is the 1,4 bond right here in between the 4th carbon in the glucose molecule and the 1st carbon in the next glucose molecule through this alpha bond. Right. And again, the enzyme you have in your saliva, this is the bond that it is cleaving when it acts upon starch. All right, alpha 1,4 right there, alpha 1,6 right there. And starch is used as a thickener in many food products. The one we're using is the shelf-stable pudding because it's cheap and easily available locally in a wide variety of flavors. And so again, we just buy these snack packs and have people originally in my class lick the spoon. So this was the original pudding. Again, you can see a very thick pudding on the spoon versus once they have licked the spoon and stirred it for a little while, you add just a little bit of this saliva. And amylase is that potent. It's like the amylase enzyme, it is good at its job. So just a little bit of saliva with the amylase, and you will liquefy the pudding in a matter of minutes, if not seconds for some people. So I'll show you some of that here in a little bit. Right. So, but again, adding the amylase to it makes it become more liquid. So we're considering maybe we should call this like a saliva liquefaction assay or something like that. I've been just calling it the pudding assay because that's easy. But I mean, what it's really doing is just it's taking what was a gel structure from that starch, cutting the starch into tinier pieces so it can no longer form a gel, and you end up liquefying it. And so again, working through some of the COVID times, we decided to develop another remote assay. And again, also thinking toward the future of trying to sort of diversify our subject population and be able to bring more people into the research. We wanted to see if we could sort of standardize some sort of assay for looking at how saliva digests starch, looking at this amylase activity. And so the framework that we decided to work within is actually something that was done by oh gosh, I've got to get the acronym right, the International Dysphagia Diet Standardization Initiative, which started, I mean, this came out in 2019, but they've been working on this for a long time before that, actually. So this is a group that basically was trying to address more from a medical perspective, making sure that we were using consistent terminology when we talk about the thickness of certain modified foods. So a lot of people if they develop trouble swallowing, you have to basically cut up or puree their solid foods. So that's the top triangle that's pointing down here at the top, or you have to thicken their drinks because it makes it easier and in theory safer for them to swallow. The thickening of the drinks is becoming a little bit less standard, but this was the practice, at least at the time. But it was really hard to get consistent information on just how thick people were making stuff. And there's a whole field about like, you know, the variability in that and how thick the drink should be and all that. But what they came up with was a method to measure it, basically to measure the thickness of a liquid without having to have any sort of fancy equipment. And so their method is basically you take a 10 ml syringe, you stick your finger over the bottom, use another 10 ml syringe to squirt 10 mils of the fluid into the syringe opening here. Then you remove your finger, and after 10 seconds, you put your finger back and you see how much liquid has flowed through the syringe. Right, so really easy to do, pretty standard size device. Most 10 mil or 10 cc syringes are pretty much the same diameter in most medical environments or supply chains. So it was a way that they could standardize the measurement of viscosity for these liquids. And I thought to myself, well, I can squirt pudding in there and we can measure how far the pudding flows through the syringe. And that would give us an indication of how much digestion somebody's saliva had done on this pudding. And so we tried that. We again did this study. The study had people consuming high starch diets and low starch diets for a couple weeks. And we were trying to see if their saliva changed over the course of that. And that didn't work out for a lot of reasons. Saw some interesting patterns with how like hard or soft they thought the foods were changing, but not anything useful related to this part of the study. But this part of the study in and of itself, I thought was pretty cool. So we had people chew on a piece of wax for 30 seconds, collect all the saliva, suck it up into a syringe and take a picture so that we could get an idea of the volume of saliva they had generated. We then had them squirt that saliva into a pudding cup, stir the pudding cup, stuck up 10 mils of that saliva saliva pudding mixture into a syringe. Again, you squirt it into a second syringe here, and then you let it flow through that syringe for 10 seconds and measure how far it's flowed. That's the whole premise of this sort of assay that we were working on. And so our initial tests were just really wild again in the variability of this. And I kind of suspected this would be the case again, because I've been doing this in class for so many years and could see the pictures of what my students had submitted for their homework. But, hadn't done it in a way where we had quantified the amount of pudding flowing through these syringes. And so these are an example of 3 people that I've kind of added this tan bar to it so that you can kind of visualize how much of the pudding saliva was left at different time points, right? So all of these samples are coming out of the same pudding cup that had the saliva added to it. And so we're taking a measurement at 2 minutes, at 4 minutes, at 6 minutes, at 8 minutes, at 10 minutes. All right, so these are repeat measurements, taking more pudding saliva mixture out of the cup over time. And, over time, it's going to become more and more liquid. But the crazy thing is that at 2 minutes, we had some people who already the pudding was completely liquefied by people's saliva. Again, this is just whatever you've generated in 30 seconds of chewing. So completely liquefied at 2 minutes versus people who basically it had no effect. The pudding didn't flow through the syringe at all. 9.8 milliliters is probably within the error of trying to suck up 10 mils of pudding and squirt it into another tube. So by the end, yeah, pretty much everybody's pudding flowed all the way through after 10 minutes. But earlier on, there's this huge variability in just how liquid people's saliva has made the pudding, right? So keep in mind, more pudding remaining means lower amylase activity, less pudding remaining means higher amylase activity, because the more liquid this gets, the more active your amylase was. And so again, really wide variety in this. And if you then take a step back and you think about it in terms of, well, what does that mean about the way people are experiencing their foods? Because foods stay in your mouth for 30 seconds, the 30 seconds it took us to generate the saliva that we were using this test. You have food in your mouth for 30 seconds often when you're chewing or swishing or things like that, depending on the texture of it. And so the person whose pudding has completely gone after 2 minutes versus the person who still has it all in the tube could be super different. Oh, look, I did include it. I wasn't sure I was going to go into all this detail on this particular one. In fact, I think in interest of time, I'm not going to talk too much about what we had people eat because not a whole lot happened. But again, the thing I really want to highlight here is how variable people were. All right. So again, we tried to do this dietary starch intervention. That didn't work really well. It didn't, cause people's salivary alpha amylase seem to go up or down. If anything, in general, having people in the study seem to reduce their salivary amylase activity. But looking at all of these dots is what I really want you to look at. So this is the amount of pudding remaining in that tube at our time points. I believe this one, let's see, we did measurements at 30 seconds and then at 2 minutes and 4 minutes in this study. So the last slide I showed you was at 2 minutes. And yeah, that's kind of a long time. You're probably not going to chew food for 2 minutes. Not all food anyway, but for 30 seconds, absolutely, people will chew food for 30 seconds. And look at this variability that you have all the way from there is absolutely no pudding left. It has been completely liquefied and flowed out of the tube to pretty much all of the pudding is left and nothing has been liquefied. And again, feeding people a lot of starch versus not feeding them a lot of starch didn't really seem to have a whole lot of effect. We might have had a little bit of a trend here on the low starch week, which is not what we were expecting. We thought, if anything, that your saliva should adapt to having more starch. But in the end, we were probably using the wrong types of starch in this. There's a whole bunch of reasons it probably didn't work. Interestingly, our low starch foods tended to be harder, so people probably had to chew more. So it's quite possible that anything we saw was actually a result of changing their chewing behavior. But anyway, nonetheless, the crazy part is really the variability and how broad these measurements are. And if we run this sort of correlations within a person, it's actually pretty consistent, like which people are these bottom dots versus which people are the top dots. So people have good correlation within themselves in terms of how quickly their saliva was liquefying the pudding, even if it didn't seem to relate to anything we were trying to do with the research study. And again, this was just the point I was making a second ago, like the low starch foods tended to be pretty hard in our study. So I'm not going to go into that. It's just too messy. The study didn't work. Basically, if you feed people a bunch of starch, it didn't change. At least the kinds of starch we fed them, it didn't change their amylase activity. But the amount of amylase is wildly different from one person to another. And so we still have to sort of validate this assay against some sort of standard commercial amylase activity kits. The only reason I haven't done it is because it just seems like, well, yeah, they're gonna correlate, because this is a very easily measured physical phenomenon. And so we'd like to do that just to show whether we can sort of quantify the amylase this way rather than just, saying, hey, you had a milliliter of pudding left. We'd, in theory, be able to actually relate it back to sort of the units for amylase activity. And we've done a little bit of this test repeatability and within subject correlation over time, because we have that study where we tried to do an intervention, but the intervention didn't change anything. Well, if the intervention didn't change anything, then we've got some repeatability measures, and it turns out it's pretty good as far as with the within subject correlations of this. And then, I've thought about, like, could we standardize with other pudding thickened products so that, if we wanted to do this in different cultures that may or may not have this particular food product, or if it varies regionally or something like that, we could standardize it. But also, I think it would be the other part of this is relating it again back to that human experience of the flavor of these foods and the texture, in this case, of these foods. And, the difference in what someone is experiencing, even when we think about, chewing a piece of bread, for someone whose saliva is going to liquefy pudding in 30 seconds versus somebody whose saliva took 10 minutes to barely have an effect. I mean, that's got to be a wildly different experience, which could then relate back to things like how quickly people eat their food or not, or how, much effort it takes them to chew their food or not. And all of those things those other aspects have been tested in relationship to, what types of foods people tend to eat and the healthiness of their diet, so to speak. And so that has a lot of potential for the future as well, I think. And so the last one I wanted to talk about here, relating these sort of flavor experiences that people have back to what is going on in saliva and whether that changes over time, gets into saliva's ability to bind bitter and astringent stimuli. All right. And so we've known for quite a long time that saliva contains proteins that can trap some bitter or astringent molecules. And particularly, a lot of this work has been done with polyphenols. And a lot of sort of the in vitro work has really been done in the wine industry. Because when we think about certain types of wine, things like Cabernets that have astringency to them, sometimes people don't like that sensation. It depends on who you are. But that sensation is also often perceived as unpleasant. And so sometimes producers of certain wines might want to have their wine be less astringent. And so there's been a lot of work done on that. Also, the cranberry industry has always been very interested in astringency for similar reasons, that, those sort of drying or rough sensations in the mouth are often perceived as unpleasant. But again, a lot of this is going to be driven by compounds like polyphenols. And so here in the purple dots, these are representing, this is my easy drawing for polyphenols here. And usually these things would bind either to surface bound proteins on the tongue. There's some mucins that get bound to the surface of your tongue. And when the polyphenols come up and bind to those proteins on the surface of your tongue, it can make it go like that. So that's the astringent sensation, where it puckers together. So that sort of condensing of those surface proteins can actually pull your tongue together. And that's part of why you feel that dry or rough sensation on your tongue. And then also a lot of these molecules can actually bind with bitter taste receptors. But again, the presence of certain proteins, such as PRPs, which are proline-rich proteins in your saliva, these can actually bind the polyphenols in solution in saliva, so that they actually don't have the opportunity to cause astringency and probably bitterness, though for some reason we tend to talk about astringency and not bitterness for these. But it's all the same classes of compounds. They differ in size a lot, but the phenomenon are similar. And in theory, these can also block bitterness as well, these salivary proteins. And so, again, a lot of work has already been done in the wine industry, sort of in the glass or in vitro, thinking about, ways you can get these polyphenols bound up into other proteins. And so in wines, we would see aggregates of proteins binding with the polyphenols in a wine as a cloudiness. Eventually, this cloudiness would precipitate out of the wine. And so you can have this happen if a wine is just basically if it has too much protein in it, or if during the processing it somehow got too much protein into it. And so that creates this cloudiness that people generally don't want in their wine. And so that's, again, where a lot of sort of these studies of the mechanism of where are these polyphenols going and what are they doing and what is making this cloudiness appear, that's where it came from. But it turns out the same phenomenon happens when you mix those polyphenols with saliva. And in saliva, what happens is people whose saliva gets very cloudy. So I've got sort of some images of what this looks like on the right-hand side of the screen. When I talk about high haze or forming a lot of cloudiness, that would be this more opaque sample all the way over toward the right-hand side of the screen. Whereas low haze, that's going to be clearer mixtures. And so people whose saliva became very hazy or very cloudy when it was mixed with tannic acid, which is a type of very large polyphenols, sort of a big oligomer of these polyphenols. People whose saliva got really cloudy, indicating there's a lot of binding going on, they're on the bottom in the squares here, they experienced less astringency, so less of that sort of puckering of the tongue than people who had a lot, or people who had a very clear samples, low haze. So the people experiencing a lot of astringency have low haze. That means there's less binding of those polyphenols with salivary proteins going on, versus the high haze have more binding of salivary proteins with the polyphenols going on. And so again, it seems that the presence of certain types of salivary proteins might actually make people experience sensations like bitterness and astringency from things like polyphenols very differently from each other. So again, another sort of individual component of your saliva that could drastically shift the experience you are having compared to the person sitting next to you in terms of how it feels when you taste these things. All right. Now, the really cool thing that has been done in rodent research, so I have a colleague, Anne-Marie Torregrossa, up at SUNY Buffalo, who collects saliva from rats. So you can imagine that's a very tedious process. But in animals, it seems like that these salivary proteins definitely block the bitter activity of molecules. So basically, she puts animals on a very high. She's done it with quinine, she's done it with tannic acids. So quinine is an alkaloid, but also very bitter. Again, as I mentioned, talking about tonic water before. And basically by feeding these animals these diets, she's able to upregulate these specific proteins in their saliva. Haven't been able to replicate that in humans very well, but at least there's some mechanistic data indicating that the more of these sort of things, quinines, polyphenols, bitter stuff you consume, the more of these types of proteins your saliva will have. And so what she's been able to do is measure a nerve response. So the cortotympany nerve, which goes from the front of your tongue up to your brain through your middle ear. So she's been able to measure in the rodents this cortotympany nerve response for taste of bitterness. And she's been able to basically create this saliva with lots of these proteins by feeding animals really, really, really high in this case, quinine diets. And then because she's working in rodents, she can take that saliva from one rodent and flow it over the tongue of another rodent while measuring these nerve responses. I have not yet figured out how I could take saliva from one person and give it to another person to measure their taste responses. I've thought about it, haven't gone there yet. But anyway, what she's basically able to show is that without protein, the animal has a higher nerve response for bitterness compared to with protein in the saliva. So if you filter the proteins out of the saliva, the animal experiences a higher nerve response, indicating more bitterness, presumably, compared to without the proteins being filtered out. So again, the proteins present in the saliva seem to be protective for bitter taste. I know we're getting toward the top of the hour here, so I'll try not to zoom through this too fast. We were trying to do some more measurements of whether, these sort of properties of the saliva contributed to the differences in taste that people experience. And so we wanted to see if we could correlate this sort of cloudiness forming in people's saliva with bitterness or astringency in actual foods. And we decided to do some beverages, some juices, particularly green juices. So we picked basically as many commercial green juices as we could find out on the market in a reasonable quantity, and that seemed pretty standardized. And then we had some fruit, some berry juices, so some cranberry, which we actually had dilute because the pure stuff is so harsh, and some fruity acai berry kind of juices. And then we also had people choose some chopped kale and chopped Brussels sprouts as well. All right, so we chopped that up and we had them chew it, and we had them rate astringency and bitterness and then correlated it back to this sort of cloudiness forming in their saliva. And the short story is you get a great relationship for sourness in cranberry juice, which is sensible. Cranberry juice has a lot of polyphenols, but also just the sourness in general of this might relate to some things getting bound up in the saliva or precipitating out based on the pH problems. But when we start to go into some of the other juices, like the relationships are a little iffy. Weirdly, we got a lot of relationships with less sweetness, with more of these proteins kind of showing up in the saliva. So on all of these charts, I'm showing you basically as you increase along the x-axis, that means it's more cloudy. That indicates that there's more of these proteins present. And if you increase along the y-axis here, that means it's a stronger sensation. And all of these plots, you're going to see they go down if they go anywhere. So in general, more protein related to less sensation. But the fact that that happened for sweetness, potentially, as well as bitterness or sourness, we were really caught off guard for that. And so it seemed to be juice specific. So here we see V8 healthy greens. We had relationships more close to relationships for several sensations, but not so much for some of the other juice blends. So this was a local store's in-store made green blend. Some of the other commercial products had absolutely no relationships. So this is a berry blend. This is another green juice blend. And so the real short version of the story here is that this may not be very consistent. This might not be a generalized effect where, certainly we're not seeing consistent effects of less bitterness with more of these proteins being present in the saliva. And it also could be really specific to the individual ingredients in these individual juices. So if there is a pattern, it's probably related to very, very specific compounds, which may or may not be very consistent from batch to batch in some of these juices. All right. So again, we didn't expect to see relationships with sweetness, but we did. So that was weird. I can't explain that. I don't know why you would experience less sweet with more of these salivary proteins. That is not the way this should work. If anything, if you're experiencing less bitterness, the sweetness should seem greater because that's the way your brain tends to interpret those mixtures occurring. So doesn't make sense to us. So I don't know if this relationship is really happening the way we thought it was originally. We certainly can find patterns in some foods that, the presence of these salivary proteins, presumably binding things like polyphenols that are bitter, might influence the way people experience the taste of them, but it's kind of a mess. to be honest. So again, this could be very ingredient specific. It does seem that some of the samples where we saw more strong effects as far as bitterness or even the sweetness go, those tended to be the samples that had the kale juice in them. So that might be worth pursuing. Kale is probably the green vegetable amongst all the samples we tried that would have the most polyphenols specifically in it rather than other types of bitter compounds. And so potentially this relationship is correct. It's just not very generalizable to broader foods rather than specific ingredients. And so again, you know, we might someday be able to develop another sort of assay kind of based on this, but it really might be very specific to very individual products or ingredients. It might not broadly explain people's flavor experience from the foods they're consuming. And so that one, I think that one needs a little bit more work. The other two I talked about today, those we're pretty excited about. They're pretty cool. But this one, it's a little hairier, which is interesting because there's actually way more research on this relationship than there is on either of the other ones in the science. All right. So thinking about sort of measuring the way your saliva works, relating it back to flavor and diet and experience, there's a lot of potential here for using applications of this, observations similar to this, to understanding how people are experiencing the flavors of their food. But the flavor experience is complicated. We need to assess how reliable some of these salivary measures are if we really want to use them to understand people's experience of flavor. If we want to predict the experience of flavor, we need kind of more consistent information. But again, these first two in particular, thinking about the way saliva could strongly be influencing the experience of fatty taste, fatty textures, even based on this emulsification assay, understanding the differences that people would experience in the texture of something that's got a lot of starch in it. You know, that relationship I just think is fascinating, how very broad, especially these two phenomena are in the way people's saliva behaves, and then the potential for that in understanding people's experiences of foods is just, to me, it's just wild. Like I was really surprised at the breadth of responses we saw on that. So some interesting potential there for helping us to understand what people are experiencing from their foods, and then also how that might change over time. So with that, I will wrap up, and I'm happy to answer any questions.
Any questions?
Can you all hear me?
Yes, we can.
Yes.
Marty Campbell. So you mentioned several times that you did this sort of without planning to in the midst of the COVID epidemic. And so one of the thoughts that I had was, and I don't know for sure whether you can reveal this or not, but did you ask your participants whether or not they'd had COVID before they participated in these different
So
studies?
you know,
Did you see any differences there?
especially that first year, I found myself a sudden weird expert of weird things, because COVID has the potential to influence taste and smell as well. And
Right.
so all of a sudden, the questions we asked to like decide who gets to be in our experiments, I'm like, all right, so some of these questions we need to be a lot more careful about making sure, like, because before in the olden times, we would just say, do you have any problems with your sense of taste and smell? And people would say no, and they'd be in the study. But now we actually test for that. And so I think in all of these studies, except for the one with bitterness, because that one we did before we did that one in the olden times before COVID was a thing. But all the other ones, we do a quick assessment of taste and smell. And it's
Okay.
actually been quite surprising to me that we probably get about 10% of our people have some evidence of taste and smell dysfunction. That or they're doing the directions wrong. I'm not sure.
Yeah.
Because we're not
Sure.
using the biggest, the expensive full sort of medical versions of the test. We're using condensed versions that are easier to do for research because money.
Right.
But so we do that. We also, people have to be healthy at the time they are doing the experiment.
Mhm.
But yeah, they could lie to us. Like they could always lie to us about these things. And so I think to me, the most useful bit has been learning to like, all right, we actually have to test people's taste and smell. We can't just ask them, hey, are you good? Because the prevalence of that has either shot up a lot, which is totally possible in the last few years, or is
Mhm.
just something that we have to watch for now and we didn't before. So
Right. Yeah. The new variable that you can't do anything about.
yeah.
Thank you.
Uh-huh.
You have 2 questions in the chat.
Yeah, I was just reading the ones in the chat. So yeah, we still are working on some things related to this research. A couple of the papers are out now, and I've of course got grants submitted and waiting to find out more. The starch one in particular, I'm hopeful for that we're going to get some more projects going on that soon, especially because like as like a food scientist in particular, there's so many interesting questions you can ask about the starch too related to this, like the ways you cook the starch, the types of starches you select, the botanical sources of the starch, all dramatically vary how well things like amylase can get to their active sites, to the substrates that they need. And so the variability of that based on the food and based on the way the food is processed could be really different. It could really change how active somebody's saliva is on a food based on the processing. So we're hoping to do some more work along that lines. And then the fat one, I'm really hoping to do some more work along the lines of the bitter one. I would think that there's more potential for the bitter one, but like I just can't get the bitter one to work out in humans the way that it seems to work out in animals. So I might shy away from bitterness and start thinking more about starch and fat. And let's see, the other question was about, are you trying to produce a product from our findings to make food more palatable, or just trying to understand people better? Maybe is the answer to all of that. So we're definitely trying to understand people better. But some of the things we learned from this might be useful to the food industry to change the way the products are formulated or processed or something like that, that would make the food better. A lot of this comes to this idea that there's this huge variability among people, and that means that there's segmentation in the market. So if you put the business hat on, variability among people means consumer segmentation. And so it's quite possible that there might be sort of subgroups of people who would really appreciate a different type of product that doesn't exist yet, that could potentially be optimized based off of some of these, sort of intrinsic phenomena. But we have to figure out whether or not they're really, whether it's stable, whether it's consistent, things like that first. So.